Good afternoon, everybody. Welcome to today's QAAFI Science Seminar. It's my pleasant duty to welcome everyone to this seminar from Associate Professor Daniel Cozzolino. Before we start, let me acknowledge the traditional owners and their custodianship of the lands on which we meet today and pay respects to their ancestors and their descendants. Daniel will give a presentation. He expects it to last about 40 minutes and that we can then have a Q&A session after that. So if you'd like to ask a question, please be thinking about it and type it into the Q&A tab at the bottom of the Zoom screen, and we will get to those at the end of Daniel's presentation. So without further ado, it's my great pleasure to introduce Daniel to you. In these strangest of strange years, Daniel joined us in January and has lived through the whole experience as a newcomer to QAAFI. So please make him welcome when you get a chance to actually meet him physically. I know he's got around and met many people already. We're absolutely delighted that he's part of the CNAFS team in QAAFI because he brings a wealth of experience in applying sensing technologies to the agri-food systems, and he's going to give us a bit of an overview of that today. So please, Daniel, take it away in your presentation, the value add of sensing technologies to agri-food systems.
Thank you, Mike, for your kind presentation. And before I start, I also like to acknowledge the traditional owners of the lands of which we are meeting today and pay my respect to their ancestors and their descendants. Today, what I would like to do is provide you with an overview about the use of sensing technologies in the agro-food systems. And I just choose to select some examples of what I've been doing before. And what I was thinking about this presentation was to provide with the capabilities and the ability that we have here in QAAFI to address these issues about the digital agriculture, digital food, and so on. I think the future of food, like it or not, is around the use of sensors as well as mathematics. As well, we need to understand the chemistry and the physics, but mathematics and sensing technologies are going to become part of what we are going to do in the future in the food sector as well as in agriculture. And again, in that link between the food and agriculture. I will provide you with some sort of background first. One is why the sensing technology is very important, and I didn't base in any specific sort of references. I know the decalogue from agriculture and the Association of Engineers and STEM are working on that as well, but I took this from the NIRC, that is the National Research Council in US, and they were talking about what are the challenges and opportunities in the future of the agro-food systems. And they were putting that in two main things. One about the challenges, how we develop high quality and nutritious food, how we are addressing issues about the food and integrity and safety, and around what is the big banner that we call about the food security, and also climate change, and how climate change will affect either the composition or any other aspect of food. And following on that is the opportunities that we are seeing now in the sector across agriculture and food. And everything that I'm going to say today applies either to agriculture and food, and that link between the agro and food systems. And the opportunities is about sensors and omics. And Mike, the other day in the QAAFI presentation, he was saying about to measure things. One of the important things that we need to in order to understand and manage what we are doing, we need to measure things. And omic technologies and sensing technologies are allowing to do that. The important part is about how we integrate these systems, and that's where people will use the things about this machine learning kind of approaches, the big data, and that is how we marriage the use of the sensors with the data systems through the big data and machine learning kind of support tools. And the other important aspect is about how these technologies are bringing together that multidisciplinary or transdisciplinary approach in the research. And again, that is something that I'm not saying anything new, but that's how probably the agro-food systems are moving into the future. And particularly the use of the sensors and big data is really making a big change in the way that we are dealing with things. You can see here, this is from one of my friends in Denmark about the classic approach of doing research, about how we need to do invasive, taking the sample, make things very slow, how this use of some methods of analysis are environmentally not good for the environment, how we are just looking one variable at a time, and looking at what we call just breaking that covariance. And on the other hand, the use of sensing technologies are allowing us to do many other things that we couldn't be doing before. And one is to use remote sensing, for example, use hyperspectral cameras, infrared instruments in drones, but also we can include and start adding many of these sensing technologies across the whole value chain. And this is very important from one of the things that we want to address here in QAAFI and particularly in CNAF. They are non-destructive, they are very, very rapid. That means we are using light and they can be rapid. They are called, or they are around the green technologies or environmentally friendly technologies. And they are really producing with this massive kind of data that we call big data, but they are multivariate in nature. But why these sensing technologies are important? Why is important? Not because Daniel or anybody else wants to do sensing technologies. It's something that is even beyond that. Is the industry is very keen to use these technologies because they can really add value in what they are doing. And this is just a very simple example of how the use of these technologies are helping the industry, particularly in terms of the quality control. They tend to reduce the cost of analysis. This is a table that came from a company in 2009, and you can see there in red the amount of money that we are, or the companies are saving just by using these technologies, and the running cost per year. That means sensing technology is not just putting a sensor and monitor and generate data. Sensing technology means that we can really monitor what happened in the value chain. We can monitor what happened in the farm, but also saving a lot of money. And that's one of the main attractive issues for the industry. What is important to know that sensing technologies, and that is hyperspectral, UAVs, cameras, many of the things are based on light. And we are using most of the sensing technologies are based on spectroscopy. And on the other hand, and together as a marriage, we are generating this massive amount of data. That means the use of sensing technology is coming together hand in hand with this big data explosion, with this generation of things like machine learning that you will hear around in many places. That means that's how sensing technology and how that sensing technology is disrupting into the food value chain and is disrupting into agriculture. And sensing technology is based on spectroscopy, is based on measuring molecules. That means what we are measuring when we are using a spectrophotometer, that light generates different levels of energy. That level of energy excites different bonds in that molecule. And these bonds produce what we call the spectra. That means sensing technologies produce a spectra that will collect all the information related to the chemistry and some other characteristics of the sample. And this is very important, and that is a very key and attractive thing for the use of these technologies. We can monitor changes in the chemistry, we can monitor changes in the process of the sample just by using this kind of sensing technologies. But what is very important is when we are looking at the whole application is sensing technologies is not just about the sensor, it's not just about the machine learning or the mathematics, it's about how we put everything together. That means application of sensing technologies implies that we need to understand the spectroscopy or the sensing technology, what happened about the sample, and about the sample is about the biology, the chemistry, anything else that happened to the sample, and also the mathematics. That means sensing technology is not just if I use a specific sensing technique or not, or it's not just about the use of machine learning. It's how we put that together in order to make an application. And that is a very important concept. And that is where many of the application of sensing technologies fail, because people, they tend not to understand how the whole complexity of that. But at the same time, if you are seeing this picture, this slide, you will see how these sensing technologies are a very good platform to make multidisciplinary or interdisciplinary research happening. And I was very proud and very lucky that in many things that I was doing in the past years was involving working with many interdisciplinary works, because we need to really make happen things in terms of application, and we need to be sharing that information with the chemists, with the biologists, and so on, in order to really better understand that application that we are developing. One of the main applications of sensing nowadays is about the food provenance, food security. And what I'm going to talk today is very briefly about some of the applications in terms of the food provenance. But food provenance, and I put just some definitions there, is not only about the geographical origin of the sample, it's about the farm production system, the cultural practices, many other aspects that make that particular food belonging to a particular region or geographical region or a particular site. But at the same time, with the provenance, the food industry now is facing issues about adulteration or fraud, substitution, and as well as authentication. And that is the things that more or less infrared spectroscopy, among other techniques, are helping the industry to better understand and to better targeting issues related mainly with food fraud and adulteration. And that's how the industry also is moving in order to use these technologies in order to targeting provenance. And of course, provenance and all of this can be measured with other techniques. But again, sensing technology allow us to go into the farm, allow us to go into a ship, they allow us to go into a truck. I mean, it's going to be very hard to put a GCMS analysis instrument inside a truck, but we can do that with an infrared instrument. There are some negative aspects of provenance of these things about food security. Around 90% of the food during 2018 and 2019 was considered and published by the Federal Drug Administration in the US that was prone to be with somehow of fraud or adulteration. That means it's a lot. It's 90% of the global food somehow is being adulterated. And even worse is the cost for the industries around, again, for the Federal Drug Administration, between 30 to 40 billion dollars per year is the cost for the industry in order to target and deal with issues associated with food fraud or food authenticity. And more recently, and one of you and some of you probably will recall the issue with the use of horse meat instead of beef meat. That became a criminal issue, particularly in UK. And you know, there are many things that are being prone to this adulteration. And that's how food now is very prone to that level of fraud or even authentication. It's very hard to distinguish, for example, what is the real salmon from salmon that was caught in a factory. Is again the issue with the horse meat, issues related with the honey. Again, 2 or 3 years ago in Australia, we were facing the issue that honey was not honey. In Europe and in US, 70% of the olive oil in the market is considered adulterated, and so on. That means these are the negatives of the provenance, to put it in this way. And again, in the UK in June, July this year, they defined with the Food Standards Agency to implement this task force, the task force that is related to food forensics, particularly associated with the police. That means now food is becoming like drugs, and particularly the issue of food fraud is becoming as important as selling heroin or methamphetamines in the market. And that's how the UK police and the Scottish police are putting these kind of task forces in order to target that. On the other hand, the positive ones, and that's where provenance is very important, and some of the things that we were discussing with Mike and some of my colleagues in CNAF is provenance is going to provide us, and particularly Queensland producers, with a very good way to differentiate and make value of what they are producing. And these two tables here present a way how in Europe they get more money in terms of the average price when they define these premium products based on the provenance. And you can see how a particular food, this is oil from Italy, Greece, or chocolate from Germany, can be paid more just to state the provenance of that particular food in the label. And the same for particular foods, that is for cheese, spices, particularly saffron, in the label, and we can authenticate that saffron came from particular region in the north of Africa. The consumers are willing to pay up to 111% more than that particular price. And that's why provenance become one of the main issues nowadays around food, because it's very important how we assure the consumers how that particular food is, where that particular food is coming from. Another thing that I'm participating is with Jasmine and Heather and other students and people around this, is about how we can help to understand uniquely Australian foods. And again, how the provenance is collecting that about the protecting the value, but also how we better understand about that particular food is coming from a specific culture, making communities more resilient, and so on. I mean, that is also what part of the provenance means in terms of us characterizing that particular foods. And before giving some examples of what I was doing before, that is something like some of my colleagues in Spain are doing nowadays, and this is from the Iberian ham authentication, and that's how is the level of sophistication that determining food provenance are becoming in Europe. This is, you can see there, operator looking at the fat using an infrared machine. that infrared machine or sensing technology, sensing and sending information to a cloud. That cloud contains information about the origin and the quality of that particular fat related to the ham quality, and how the consumers at the end can have an app to distinguish or knowing from what a particular ham was coming from, as well as the quality of that particular ham. And there's another application that is from a company that is selling instruments, how they are now looking at applications about that, adding that value in that value chain about the use of sensing technologies. And here is an example of how you can target about the composition related to the health, if they have high sugar content or not, or if they have, for example, in terms of the authentication, if a particular batch of milk has high concentration of melamine in the milk or not. And I'm going to provide you with very general examples of what I was working the last 10 years before coming here into QAAFI and COVID environment. We were working on with some of my colleagues in Ireland about looking at how implementing these technologies in the fish value chain, particularly about how detect particular things in the fish to confirm if that particular fish is that fish or not, if we can classify from different groups and also identify. And I think that's very important because during the weekend I was listening to the ABC and the barramundi producers in Queensland and in Australia in general, they were really worried about how they can identify and trace and make the consumers in Australia realize that barramundi here is from Australia and it's not coming from Vietnam or any other Asian countries. That means this is very important. That's very important in order to add that value to what the producers here in Queensland are doing. I was working with wine, and this is just an example that we were able to identify organic and non-organic wine. That means normal wine versus the organic wine. And there was a very interesting exercise that we did with two of the wineries that produce organic wine in South Australia. And in this case, we were applying mid-infrared spectroscopy, and we were able to identify samples that were considered non-organic because they come in from a patch, from a vineyard that was that particular vines were produced under traditional cropping systems compared with the grapes that were coming from organic conditions. And there is even late at the beginning of my work with traceability, there's a work that we did back in Uruguay, in South America. Uruguay was trying to sell meat as characterized as a natural meat and compete with Australia, Argentina, and Brazil. And we have this experiment when we fed animals with pastures and silage. We were scanning the carcass and the slaughterhouse, as well as we're scanning the longissimus dorsi from the steak. And we were able to identify that just the origin of that particular piece of meat or carcass based on the information provided by the sensor that we were using. In this case was a NIR sensor. More recently, and regardless COVID, we were doing some work, and this is, I need to acknowledge here Eshetu, a PhD student working with Jasmina, as well as the indigenous communities in Western Australia, that we were collecting these samples. That's from Kakadu plum. And we were collecting from the whole Kakadu plum the spectra, and just based on the spectra and doing some big data machine learning kind of approach, we were able to identify the origin of that particular fruit, if we're coming from region A or B. and region A or B were related to where that particular samples were collected. If they were close to the ocean or they were more inland. And we were able to identify that based on the signals that were recorded in the spectra. And that is the continuation of this project. Is based on some funding with the CRC for the Northern Territory, and using that machine that we have here, that is our NIR sensor, that we can look at quality as well as authenticity of provenance in the food. And that is something that is very interesting, and the communities that we are working are very excited because they are seeing how we can add value to this particular fruit just to monitor not only if they have high or low vitamin C, but we can add other layers of information related to the provenance that can make really them get much better prices when they go to the market with these particular fruits. And that's more or less that how we have our brainstorm, and that's how we are seeing the future, and that's why we are thinking about that adding value into the value chain of the Kakadu plant, using these kind of approaches, how we can look at the fruit in the trees, how we can characterize and individually target specific fruits in the field using this kind of sensing technologies, sending that technology to a big cloud where we have access to the information in terms of keep doing the research that we are doing here, or providing that information back to the communities to use as a way to improve the way that they are selling the Kakadu plant or any other products they're developing. There was a little bit about provenance, it's just in a very quick nutshell. And the other thing that I want to show you what other things I was doing, and I put this kind of very marketing name about biospectroscopy. Again, people use sensing technologies and developing a lot of jargon. That is biospectroscopy, I think looks very cool, but it was something that we were starting when I was working in Adelaide with the Barley Breeding program. We were looking at how we can analyze single seeds using spectroscopy, with the same concept of that spectra is providing us with a fingerprint about the sample. This is one of the instruments that we are using. It's a very small instrument. We put the barley grain there, and we were obtaining the fingerprint. And the fingerprint is that kind of a spectra where we can, we know where the lipids, where the proteins, the carbohydrates as a main groups of compounds, but we can also even go deep in understanding what happened with particular esters groups or particular carbohydrates or components of the polysaccharides like amylose and amylopectin, because they can have specific signals in the infrared. And some of you that they are familiar with NIR and infrared in general, we will always think that in order to use infrared, you need to develop a calibration. Nowadays, and with the advance of, again, a lot of big data, with a new computing systems, with new algorithms, we tend not to go with a pathway of just looking at calibrations. We can do many other things. One of the things that we are doing, and that was try to implement it in the Barley program in order to look at the new germoplasm that they were developing, we're looking at the ratios between specific frequencies in specific regions that were important for what we want to achieve. In our case, we wanted to improve the malt extract that is related to beer quality. And what we were doing we were looking at these kind of ratios between the non-carbohydrate to protein region. That means we know, back to the previous one, we know that particular frequency here, we know the frequency of the carbohydrates, and just doing that calculation, we were able to look at values that were related to specific varieties or with the controls that we were using. That means rather to do a calibration for hot water extract or for protein, we were able to speed up, make a high throughput method, just looking at those ratios in order to interpret which of those materials were able to produce high, very high, low, or intermediate hot water extract. That is the measurement of how much extraction is coming from that particular grain in order to get fermentable carbohydrate to produce beer. Some similar approaches, one of the PhD students in the IRC Training Center for Uniquely Australians is working on similar approach at Diamo, and Diamo is working on water seeds, and he's going to apply the same principles in order to understand differences between different varieties of water seeds using the same approach. And I will provide you with some of the future work or things that I have started here in different areas. One is the classic approach of using sensors to measure things, as Mike says in his presentation 1 or 2 weeks ago. It's about how we can speed up the way to measure things. And it's very important because with that we can reduce the cost of analysis. That means one of the things that I learned during COVID, that we need to be smart in the way that we are doing things. The use of sensing technologies can provide us with a tool to become more smart in the way that we are doing things in the lab, even in the field. The other area of research that I'm working is in the food authenticity and provenance, and that is together with my colleague Heather Smythe. We were working in provenance when she was at the One Research Institute almost 12, 14 years ago, and we were continuing working on that. And that is, the idea is how we can merge sensory consumers analysis with sensing technologies. And that is probably what prompted to the next slide here, that sensing technologies can also help us to get rapid diagnostic tools for animals and humans. And I've been lucky here to participate in one work with Luis and Karen and Diogo in Decathlon, looking at the analysis of hair using NIR. I've been working with Heather to look at the NIR analysis of the tongue. That means analyzing with the NIR the tongue and how we can relate that to some of the aspects of consuming or consumer research or even physiology related to sensory. And lately with one of the PhD students of Mike, looking at how we can do saliva analysis as well in order to relate that information to physiology and to better understand what happened in that interaction between food, saliva, and another aspect in terms of the physiology. And the other area that is really important is how we can really better understand changes in the composition related to that climate change using these technologies. And it's inevitable that climate change is producing changes in the protein, in the lipids, in the carbohydrates that constitute the grains, many other plants. And again, using sensing technologies can help us to better understand what happened with that effect of climate change in the commodities as well as on the foods. I just finalizing here and try to summarize one of the main things or the main frames that how these sensing technologies is becoming part of Synapse, and how we discussing with Mike is going to become part of what we are doing across not only Synapse but with other groups in QAAFI. And lucky enough that I'm trying to collaborate with Hickey, Ian Godwin, with Luis, and many other people around, many other groups across the university. That means this is one of the things that I'm really lucky to be part of the sensing technology group, because I can really promote doing more interdisciplinary research. But one of the main things that we want to do is just look at this rapid toolbox, and mainly with the concept of how we can use these technologies to protect value, or how we can maintain and maximize that value of a product. And that is very important. Not only protect, but how we maintain and maximize, particularly in the term of the consistency of the foods produced. And again, I cannot escape to the jargon, and many other people are using that word. I think is what digital food is going to be part in the future, how we can that is the use of these sensing technologies, this big data in looking at this digital food. And related to that, and related to the big data, is about that digital signature. That means now we know that the spectra can contain that digital signature of a food. And if you do anything to that food, if you change something in that particular food, either by heating or not heating, changing the storage conditions, or adding something else, that digital signature is going to change. And that is going to become very important in some area that for me was very new to see that happen, like food forensics. How food forensics, like CSI, is becoming very important in areas of in the field of the provenance, fraud, and authentication. And the other two things, just finishing here, is sensing technologies is really helping on making this agro system more sustainable. That means we can very quick monitor what happened in any of the systems. And we can use that data later in models, in making support decision tools, because we are generating data that can be used online, at line, very, very quick to monitor any change that happened in any of the systems that we are working. And that was the idea of this seminar, was to introduce a little bit of what I was doing or what I plan to do. And again, I'm here, happy to collaborate with any of you, and I'm more than happy to address any of your questions. Thank you.
Thank you very much, Daniel. That was yeah, inspiring. Lots of great examples and the vision for the future, which I'm sure people will be interested in asking questions about. So I'm just going to go back to an earlier question that was in the chat. So please, if you have questions, put them in the Q&A panel. The first question that came up was around this fraud area. So in Australia, how does that work? Who has responsibility for ensuring that what it says on the label really is in the packet?
That is a very interesting and tricky question. So far, the initiative from the federal government was to look at the labels and how they decide to say that they need to be made in Australia or with products they are based in Australia. That means that is generally what the government does, is making a recommendation about the labels. But so far, and I think that what happened with the issue with honey, it wasn't many organizations that were really dealing with that. I know that each state can deal with that, and each state, each of the Department of Agriculture or Commerce are dealing with that, but it's still not a unique front to target that. I'm sorry that I was very vague, but it's somehow that's what it is now.
No, it's a good question, and the UK initiative is an interesting one that finally recognising that there is a systemic issue here and that this needs to be taken seriously. I agree, Daniel. We haven't yet got that system in place in Australia. So running through some other questions. So one from Pat Blackall. He's going back to the provenance, and I guess he's really asking the G by E type question. So how does seasonal variation play against regional patterns? So when you talk about a regional signature, is that affected by climate, temperature, rain, so on, as well as the physical location?
The short answer is yes, and that's why the need for this kind of sort of big data approaches. I mean, it's all these provenance studies that are based on the use of these kind of sensors. They need to train the system, and train the system means you need to have a lot of data. We can complement, and that's how the approach in Europe is now. They can complement that information, for example, doing isotope analysis, and we can add that as well as another way of training the system. That means I put everything probably too simple, but the complexity of looking at provenance sometimes is beyond that one single thing. Is how we put together many people thinking and many sensors, mathematics, and so on, is how provenance, it must be target in the future. Again, it's not that single spectra is going to give the name of the dog. It's more complex than that.
Yeah. So there was a related question there, which is saying, is it possible to separate the differences in normal seasonal climate variation from regional differences? And maybe you can comment about, is there a future scenario where weather data is integrated and used to help shape the outcomes from spectral sensing?
Yes, and there is some approaches nowadays that they are doing that. I mean, again, back to that concept of that big data. I mean, big data doesn't mean that we need to have many samples. It's how good information is added into the modeling in order to explain what happened with the system. And that is how complementary technologies work, how we need to get that kind of background about the chemistry, the physics, the environment, the temperature, because that also is adding into understanding that provenance. That's what provenance is becoming a very exciting topic, because it's moving or making us to shake a little bit the brain, thinking in a different way as we were thinking until now.
Yeah. So I think the analogy here is probably in the wine sector that's got this most established. We have different years of wine. So there's a characteristic, say, of a Cab Sav from the Barossa, but it will be different in 2018 and 2019. And there's no reason why we shouldn't expect that to also be the case with our crops, but there's also no reason why we can't monitor and keep on top of that. Yeah. So the next question is from John Dixon around farm input quality. So he's asking, are you aware of any rapid tests for confirming crop seed variety or viability for germination? Or another topic, fertilizer nutrient composition?
With the seeds, I know there's some private companies in Australia that are doing that, and the same with the fertilizers. I mean, sensing technology and the use of infrared, NIR, mid-infrared, hyperspectral is being used for the last 10 years in targeting all of that. The main thing is all of that still is in the infants, to put in a way, because, and that's what is sometimes very sad, there's not many people with expertise on that. and as well as it's a lot of overselling from some companies, and that is also make things very bad for the use of the sensing technologies. And that's what happened sometimes in the past as well. But yes, there are technologies that are being used for measuring nitrogen in the soil, measuring nitrogen in the plants, as well as to look at seed vigor and other characteristics in seeds. Yeah.
Can you maybe give us some insight into the cost of the hardware here? So you showed that handheld infrared detector. Obviously, there's much more than just getting the data. You need the right modeling and so on. But just for that hardware, what's the cost now, and how do you see that going into the future?
Yeah, well, again, hardware is probably one of the main barriers of adoption. The one that we got here in QAAFI and is again used, it can be used for everybody. I mean, it's happy to share that instrument. It's around $20,000, $25,000. But the other two that I show in the pictures, the one that they use in Spain and the other one, are coming into the market around 5,000 American dollars. That means we always said that hardware is going to be cheap, and of course it's going to be cheap. But then $1,000 probably will be the floor of this kind of instrumentation. I don't think that as today we can go cheaper than that. But $1,000, yeah, is considered cheap.
So I think the message is, yeah, be thinking now, because in the future this is going to be extremely affordable technology. Yeah. So we need to be exploring what's possible now. Okay, another question. This one from Bob Gilbert. He wants to know whether it's possible to detect the amount of starch in a leaf. So there's very small amounts in a leaf and or a grain remotely. And I guess maybe you can comment about how remote is remote. How close do you have to get to something to get that sort of analytical data?
Yeah, well, thank you, Bob, and hope that you're still alive in China. How remote is remote? That is a very good question. I mean, you need to remember when you do things remotely, particularly when we use drones and cameras in drones, we have what we call that whole noise from the environment. That means these things are affecting somehow the way that the energy goes into the sample and reflecting back to the detector. And that is an issue. And that's an issue because that will define that precision of what you want to do. I mean, if you want to look at the same precision in the lab, my answer will be, it's not going to happen. But if you want to know if you have low, medium, and high of that starch or anything else, the answer will be yes. And particularly today, for example, if you look at the news, NASA found water in the moon, and using SOFIA, that is a 747 plane that they used, and they found water in the moon using an infrared instrument. But they were very highly precise and sensitive sensor that was targeting a specific wavelength that was related to water. And I think probably that needs to be the approach when we are looking at that. Just develop something that is highly precise and is targeting the frequencies that we know where starch or proteins are living. In that case, probably we can get a little bit of improving the precision of our model.
Yeah, that's a great analogy, Daniel. That the news this morning, which was just mind-blowing, the fact that you could see the vibration of water molecules on the moon from something just flying just above our atmosphere. That's, yeah, think about that, guys. And what can you do? Not that we're necessarily going to be growing crops on the moon in the near future, but think about that on Earth as well, the precision that you can get there. Okay, another question, a bit more general. I think this one from Sharam. So that what happens when you have overlap? So when you're trying to segregate, you showed organic, non-organic wine or whatever, and there will be cases where there's overlap. So how do you treat that overlap? And what are the tricks of the trade in disentangling overlapping data sets?
we have two approaches. One is the mathematical way to do that, and that's been more or less used. Similar approach is being used for detecting cancer in patients. That means we can look at the sensitivity of the model based on the true negatives, the false negative, false positives, and so on. That means we can do some calculations based on that and estimate the probability of that. The other obvious one, and that's the one that we did, we're taking the samples back to the lab, analyze the sample from things that we know that are related or explaining that origin of that particular sample. In our case, were sugars and some of the phenolics, and we were measuring the sugars and the phenolics to make sure why they were overlapping. And in some cases, the overlap was real. That means that variability, we are dealing with the variability that is natural in the system, in the food. I mean, that's how some is not that we are dealing just with a pure chemical there. It's the whole matrix, and the food matrix is beyond the starch or only the phenols. It's how they interact in the whole matrix. And somehow the sensitivity of the instrumentation put us in that very awkward position. What is true or what is not true? But again, two options. One is doing the mathematics, yes, but I would suggest go to the lab and try to understand the matrix. That is very important when we are dealing with that.
Okay, next question is, we're back to, I guess, fraud protection or prevention. So Ricardo wants to know about any comments regarding deliberately adding something as a tracer molecule that you can then pick up later and show compliance or intactness, integrity. Do you have any comments on that?
Yes, there's some, the pharmaceutical industry is good on that. They create these libraries of contamination, and they can follow back and forwards what happened with that particular product. And in the food industry, we were doing what we call this artificial adulteration. One exercise that I did with one master's some years ago, we were looking at the contamination of olive oil with sunflower, and we were doing in the lab mixtures of olive oil, sunflower into olive oil, in volume to volume kind of mixtures, just to look at where were the changes in the spectra, and then from there build a model that we were able to identify that. And that's, some of the industries are doing that nowadays and incorporating that as a quality control checks.
Okay, thank you. So the next question is about equipment again. So this is around analyzing the composition of plants in the field, avoiding disruptive measurements. What type of equipment do you think it's worth investing in to do that? For example, hyperspectral cameras, micro NIR, and so on.
Once again, I think that is what do you want to achieve? That's very important. It's not only about the instrumentation. I think it's having very clear what you want to achieve. I mean, hyperspectral is a very interesting technology, and it's been used a lot nowadays, but again, got some issues. The same with the use of portable instruments with the contact instruments. They are very good, but also they have the issues. I mean, at the end of the day, the application will depend on what we want to achieve. Ideally, and that's what people, in order to be safe, is saying, it's better to complement the two technologies. But once again, depends on what you want to do. For example, in the case of the Kakadu plum, we are seeing that the instrument that we have, that pen that we have, is good enough to achieve what the communities want. And I think starting in that way is good, because it's showing how the technology can add and can help understanding the system. And that is, is a part of that extension and translation of the research into the final user. And I'm sorry if I was very clear in my answer, but depends on what you want to achieve.
Yeah, Come and talk to Daniel. he'll put you straight. So the last question we got time for, I think, here is onto a different topic. It's about microbes, and the question is in the context of detecting organisms such as soil microbes. That maybe we can bring that out a bit more and talk about, say, salmonella in chickens or whatever. So what's the potential currently, and how do you see the future in terms of detecting specific microbes that are either a positive, like probiotics, or a negative, like disease-causing organisms?
Yes, well, it's 2 or 3 reviews that came very late, probably 2 or 3 weeks ago. One is on the microbiome, and they are saying and using in some of the applications infrared and mid-infrared and even Raman spectroscopy to look at individual species, and again trying to identify what is there. And again, you need to always be make in mind that it's like a cascade kind of research. Is what is there, if it's there or not, and then going deep and deep in the understanding. And because of that, we have different tools to do that. I mean, you are not going to go to a little screw to take a little screw with a big hammer. You will go to the little screw first, and the same and vice versa. But the short answer, yes, we can do that, and we have some applications that are trying to look at that in terms of the microbiology, identifying species and things like that.
Okay, thank you so much. I think for giving such great answers, and we've worked you very hard, Daniel, but this has been a very exciting session. I think hopefully everyone's getting the picture that there's a new era here in terms of being able to gather information about systems in our food supply chain and use it to advantage, and that there's a lot more to come here, particularly in Queensland, where our food system is very much an agriculture-based one, and our future is very much around trying to find those premiums and finding those premiums around and being able to demonstrate those premiums through the supply chain to the consumers, and justifying the prices that we want to be able to make from our products. So we see this as a really intrinsic part of that future, and one that continues to evolve. The hardware and the software and the mathematics continues to evolve here. So thank you so much, Daniel. An open invite on Daniel's behalf to go and talk to him if this has piqued your interest within QAAFI, and we're looking to see what are the best projects, what are the best ways of exploiting this capability for the benefit of the agri-food supply chain, particularly in Queensland. So thank you once again, Daniel. Thank you everyone else, and come back the same time next week for another QAAFI Science Seminar. Have a good day, everyone.
See you later.